to be here because I have been working in banana for like almost 10 years. And when Dr. Biju asked me to deliver a lecture over here, I said, okay, well, I can just summarize a little bit of my work and present it here for the research community. Well, who is not working in the agriculture field, but who is working in like botanical field. Basically, I am a botanist. So instead of actually spreading over how to cultivate banana, I think I was more interested to know the genetics of banana. So my work is concentrated on the genetics of banana. And since we are working in a crop for a long time, we will come to know about different part of that particular crop and see how actually it can be beneficial for the entire population. So my lecture is organized in such a way. I mean, I can just give you an overview about why food security is very important for us now, and then how banana biodiversity is making a major role in that area, and what are the molecular tools we can apply in this crop to improve the crop in a better way. So that is the whole area of my topic for this presentation. And now let us start with the food security. If you look into this 2030 agenda of the Sustainable Development of UN, you can see that they have set 17 Sustainable Development Goals. I am not going to describe all the goals, but you see the most important, the priority, the first goal was no poverty. So that was the priority for them. And the last item was partnership to achieve the goals. So everybody know that we have to work in partnership. Nobody can work in isolated compartment now to achieve something good for the mankind. So everybody have to collaborate and share ideas and then come to a good conclusion which can be beneficial for the human beings. So that is when I actually looked into the SDG, this Sustainable Development Goals, that is what I found, Overall, there is a kind of collaboration idea. Why this is so important, this 17 developmental goals? Because for the coming at least 15 years, these will be the national agenda for all the countries to develop their own policies in sustainable developmental area. So anything we are planning to give a proposal, it has to be somehow related to any of these goals. Then only the funding agency will give you the funds. So that is an introduction of the Sustainable Developmental Goals. And now you see, if you actually look into each and every one of the goals, you see the most important thing is that, everything interconnected there, but somehow the most important thing is that the poverty and then hunger and the climate change. So that is the thrust area among the 17 developmental goals. And these three thrust area, we can actually find solution if we really look into food and agriculture sector. If we have improvement or innovations in food and agriculture sector, we can actually find solutions for eradicating poverty, eradicating hunger, and fight solutions for this climate change. So this area, food and agriculture, is the most important area for the survival of mankind. Now I think everybody is realizing it. I just have to say a little bit of thing here, because I used to go for as an expert for conducting interview for the school children's merit scholarship schemes. The children, they will be studying in the 8th standard when they write the exam, and when they come for the interview, they will be studying in the 9th standard. So when we had this children all around Kerala, and we were conducting the interview in SCRT, they were very bright children, because the exam is very tough. It is not easy to pass that examination. They were very bright children, but every one of them wanted to do research in space science. They were not at all interested in plants. I was asking them, why the jasmine flower in the morning, and I still remember him. He was asking me, oh, I never noticed, the jasmine was flowering in night. And I have the plant in my home itself, but I never noticed it is flowering in night. But he wanted to do research in space science. So I was telling those children, why don't you look around first, rather than look up in the sky. You can do that, but at least see the plants around you. If you don't have anything to eat, how do you actually do research? So for our own, sustainability, we have to have food. It has to be cultivated in the soil. So you have to see around and see things are not working the way it was earlier. So we have to do research in plant science too. So studying plant is not a bad thing. You have to do that. Now I think people are realizing that how important this food and agriculture sector is. Now food is very important. Now if you look into the global food demand, you can think that we have to have 4 priority areas to look into it, because the most important thing is that availability of food. We can make food available in 2 ways. We can actually have the domestic production of the food, and the second thing is we can actually have trade. You can actually do the trade of this food material, so it can be available for us too. The second thing is food access. This malnutrition is not often caused because of scarcity, because it is the inability for people to access food. Even if food is there, they are not able to access food. So accessibility is the second important thing. And the third important thing is the food utilization. Food should be safe. We cannot eat the spoiled food. So it has to be safe. And also you have to know how we can cook it nutritionally without losing the nutrition part of it. So empowerment, education in the area of nutritious food is very important, so that utilization can be in the proper way. And the last but not the least, it has to be stable. That means the food has to be available all the time. Suppose there is a natural calamity, this Oki came now, right? So immediately a section of the people, they don't get any food at all. Now the government have to take measures so that the food is available for them during that disaster time. So the food availability should actually overcome the natural disasters. It has to be available throughout irrespective of the natural disasters. If we meet all these 4 important things, then the global food demand can be met properly. Now we come to the crop banana. Why banana is an important crop? If you think banana as a crop, as a substitute for cereal, it can provide up to 25% of daily calories. So that is a good crop. That is we can, instead of having rice, you can eat a banana also. At least you will get 25% of your calorie. And also, it is a universally acceptable fruit. Anywhere in the world you go, you see banana is available there, and everybody loves to eat a banana. So we can even skip breakfast by just having 1 banana. It's very convenient. It is very safe. It can be kept for a long time without even having anything, without even having a refrigerated thing. So that is a universal fruit, universally acceptable. The taste is I haven't seen anybody who doesn't like to eat a banana. So that kind of acceptability is not there for any other fruit. And then also, we can even generate income. If you look into the statistics, you can see that, even a smallholder farmers, 75% of their income comes from the banana cultivation. See, banana is a crop which has got calorie. It is it has got universal acceptance, and also we can generate income from this crop. All this data I got from the website given in the presentation. And if you look into around the world, wherever the research is going on, you can see that there is regional network well connected between the countries where the research is going on. For that matter, for Asian countries, we have the BAPNet, which is the network is connected all the Asian countries where research in banana is going on. And in African country, we have Varnesia and Innovate Panama, and also in South America, we have Musalak. All these internet connectivity between these regions make banana research very easy. We don't have to repeat the same experiment where somebody else is doing it. We get the data, all the people share the information, and the varieties are accessible to anywhere in the world where you are working. So doing research in banana is really a good thing. You don't be lacking knowledge, and you don't be lacking the information. So information is very important when we do cutting edge research. It is really expensive. We don't have to repeat so many things which somebody else have already done. So that kind of interconnectivity is there if you do research in banana. So for the young faculty who is sitting there, I think this is a really important crop. So you can actually pick it up and then start doing, because there is so many things to be done in this crop. And now you see the production of by country and by type. From the map you see India has got a very prominent role. That is the only one country where almost all the different type of cultivars like cooking banana, desert banana, and the plantain varieties, all the four different sectors of banana varieties are being cultivated only in India, for that matter. And they are the largest producer too. And we have in Brazil, we have a little bit of banana, not a little bit, majority of banana cultivation is happening, but they don't have much of these other varieties. And also we have in Africa a large sector, they are actually cultivating only the cooking banana. The desert banana is not that much there. But in all these South American countries, the majority of the cultivars are Cavendish banana. If you look into the diversity, India has got a very prominent role in banana cultivation. You all know that by 2050, the entire world population is going to surpass 9 billion. That means that the agriculture has to enhance the production up to at least by 70%. That means that we have to have innovative means. I am not actually against Dr. Jayaprakash, who is for organic cultivation, but I myself don't believe that we can meet our population demand by just simply doing organic cultivation. We have to do organic cultivation, but that doesn't mean that that is the solution for actually feeding the poor. When we are hungry, we need food. We have to have food for the food in front of those people, they are hungry. For that, we have to do the genetic engineering research and then do things where the food production can be enhanced. So I am among those people who actually wanted to do research and enhance the food production. Because of this statistics, we have to provide food for all the people who is hungry. Now, let us see the banana varieties. As I said, this interconnecting link between the banana research group, the Catholic University in Leuven, they have about more than 1,500 variety collection available with them, and anywhere in the world you are working, you can actually request for the variety or the cultivar which you wanted to receive, the sample will be provided for you. And if you do one sample analysis, they will do it for free for you also. So that kind of resource is available if you wanted to do research in banana. And then as I said, if you look into the cultivars around the world, we see about 47% of the cultivar belong to a single variety, that is the Cavendish banana. Why everybody is interested to cultivate the Cavendish? Because because the priority for a farmer, that is the higher yield. And also that is one cultivar which is less prone to damage from storms, because it is a short variety, so it will actually overcome the damage by storms, and it has got better recovery from natural disaster. And the most important thing about the Cavendish banana is the banana fruit has got long shelf life. It is very suitable, the taste is suitable for the international market. It looks beautiful, and it doesn't have the black patches developed in that banana for a long period. So that is the universally, even in south, from the south, if you go to north, I think that is the only banana. It is freely available for any of us. Now I am actually coming to the research which has been conducted in our department with my students and myself. We started the research. In the beginning, we collected 38 banana cultivars, from the agriculture from near Nedamangada. And then all the cultivars we started tissue culturing, so we have enough samples for our own work. So when we collected the 38 cultivars, even though the agriculture university has been classified it based on the variety and based on the genome, we wanted to verify whether the cultivar belong to the same genomes. everybody who is working or who is related with banana, you know that we have two different kinds of banana. Some of them are diploid and some of them are triploid. The diploid banana, we have desert banana that belong to AA genome group, and we have cooking banana which belong to AB group. In triploid also we have triploid A, B, and a triploid A, B, B. Depending upon the B content, that means the starch content will be increased, and then cooking quality will be increased. When there is no B genome, that means it belongs to a desert banana. So that is the general concept. So among the 38 cultivars, we did the genome analysis for the first time, and then used different molecular markers, and then tried to classify whether these banana belong to a particular class as reported in the literature. So the first molecular marker first actually we classified based on the morphology itself. As you see in the PCO chart, you can see that A genome, whether it is a diploid or triploid, all of them belong to one cluster, whereas other group AB, AAB or ABB or BB, they are actually a larger group, but there are many outliers, and we couldn't classify it in a different sector properly with the morphological characters only. So that is why we actually looked into the molecular marker. Now, we used the simplest molecular marker, that is the RAPD marker. And using the RAPD marker, we could clearly classify the triploid A. All the triploid A belong to one group, but AB and AAB, well, they were actually a little bit of clustering, but still there are many of them were outliers, and BB and ABB also belong to another group. So this RAPD marker was not a good marker for classifying the genomic character of banana cultivar, which we had. So we actually used another marker, that is STMS marker, that is more prominent marker and belong to the microsatellite groups, and we could actually classify it based on the genome itself. As you see in the plot, all the group can be separated well enough using the satellite marker. Now we did the OPGMA, and you can see in this in the cluster, all the diploid A belong to one cluster, and the triploid AA belong to the the first cluster, second group. And in second cluster, we have AB in a group, AAB in another group, and triploid AAB and ABB belongs to another group. B was actually in a separate line. So this was a good marker, so we can actually classify all the banana cultivar available in our own departmental germplasm in different groups using this marker. So this work was actually giving us a lot of satisfaction, so that now we know which group our banana cultivar belongs to. We did a lot of tissue culture, but I am not showing it because everybody know how to do tissue culture in banana. But I think some of the things which we got during our tissue culture part was very interesting. I am just showing you only those slides which was really interesting. One thing is that we developed an IRA primer. That is a retrotransposon based primer. Based on that primer, we could see that only those cultivar which has got a B genome, which has got a specific band. So we did sequence that band, and that band has got also a specificity. When we actually sequence that band and see the band was using a restriction enzyme ALO1, that was actually cutting in different area, whether it belonged to AAB or ABB. So that molecular marker was a kind of robust marker where we can actually see whether this banana has got a B genome at least. Whether it has got 2 B genome, 1 B genome, that was also this marker was able to classify. This was published in one Euphytica journal, and it is a kind of well sorted paper. So many people actually looked into it. Now see the tissue culture part of the work, which I already said that I'm actually saying only the interesting things we got during the entire tissue culture area. Usually everybody actually culture the rhizome or the inflorescence tip as an explant for starting the culture. We also did that thing, but along with that, we actually inoculated some bract also, the innermost bract regions, the basal part of the bract region. We did get comlet-like things from those bract. So number of comblets was not actually many, but the comblets were more stable, and transferring the comblet to the soil was much easier. So that it looked like a miniature rhizome itself. So that was really very interesting aspect of that study, and many comblets can be developed from this diploid and triploid banana, and rather than having like different stages of tissue culture, like first the shoot regeneration, then the root and acclimatization, and then transferring to the field. If we have a comblet, we can directly transfer the comblet for rooting and then to the field. So the time and the cost can be reduced much significantly if you use this technology for doing tissue culture. And another thing was the somatic embryogenesis we got from the bractex plants. The somatic embryos, the number of embryos were much higher. From a single cell itself, we could isolate a flask full of somatic embryos, and the somatic embryo was capable of regenerating the whole entire plant. And when we transferred those plants into the field, we got about 85% of survival in those plants. And we even did cryopreservation techniques so that we can actually save the plants for a longer period. And we enhanced the somatic embryogenic production by addition of biotin, ascorbic acid, and glutamine, and everything is giving a positive result. And when we did the same analysis of the callus, which we have got two different type of callus, one non-embryogenic callus and one embryogenic callus. We could see that we have primary, secondary somatic embryos, and we have if we actually enhance the strength of the SEM analysis, into 700, into 3000, into 5000, into 10,000, like that, in enhanced view, we get secondary and tertiary somatic embryos. That means this callus keep on producing somatic embryos. And now actually we can think something different. As I said, we did establish lot of in vitro culture techniques in the cultivars. Almost all the cultivar we established tissue culture protocols, and everything was transferred into the field, and we had a banana field where we had all the regenerated plants transferred, and the field was maintained in the department. But during this tissue culture time itself, when we cultivated the red banana, many of the regenerated plants were actually turned into green in color. So in the market also you can get red banana and the green variety of the red one. So they taste same, and the nutritional analysis showed that the fruit has got all the nutritional quality just like the red banana. Only the thing is that the color is green. But because of the color, naturally the market value is less. Everybody wanted to have the red banana, thinking that it is good for diabetic. Maybe that is true, because when we did the nutritional analysis, we found that the reducing sugar content is very less in red banana. But anyway, since we got this tissue culture plant, which is green in color, we were very interested. We wanted to know how it became green, because we actually cultured the red banana, how it became green during the tissue culture phase. So we wanted to see, how can, this was the question, the hypothetical question we faced, whether the red banana is a chimera consisting of red skin on a green core, or the variegation is a result from the developmental accidents due to a somatic mutation. So this was our research questions for a couple of years later. And when we see the epidermis peeling of the banana, we could see, in red banana, we could see the vacuoles filled with anthocyanin. That means that anthocyanin, the pathway is working in the red banana. But in the green banana, the peel doesn't have any anthocyanin naturally. That is why there is no color. So that means that in red banana, anthocyanin pigment pathway is working. That is for sure. So we wanted to test the genes. I am not actually showing the pathways, but when we actually selected certain genes which is very prominent in the anthocyanin pathways, the MYC gene is a regulatory gene, and chalcone synthase, DFR and ACR genes, they have got prominent role in anthocyanin pathway, and developed primers for that genes, and then did the analysis, and we got single bands for chalcone synthase. Actually, we got single bands for all the four genes, but chalcone synthase was the gene we selected because of based on the sequence data. The band was sequenced, and the sequence data showed that in this we have got 1, 2, 3, 4 lines of bands. Like, all the 4 different bands shows that chalcone synthase gene fragment isolated from the red banana, that is the last one. And then above that we have the normal green banana, and then we have got green banana and another B genome as a control. So that means that in the sequence data you can see 3 lines which has got green color, and the last line which showed the red banana. See, the difference in the sequence occur only in the chalcone synthase gene obtained from the red banana. When we do the neighbor joining tree, in the tree we can see all other gene sequences fall into one cluster, whereas the chalcone synthase gene from the red banana which belong to a different group. Now we actually converted that gene into a protein sequence and did the analysis of the protein sequences. Again, we can see that the Chn protein from the red banana has got amino acid variations in that sequence. Now, you see in the table, I have actually picked up all the amino acid variation from the red banana, belong to other green ones. And in the amino acids variations, when the alanine amino acid is because of a transversion mutation, when alanine amino acid from the green is transferred by a proline amino acid, that actually give a structural variation to the protein, because proline has got a very significant, this is a very different amino acids, and it will have a structural orientation for the protein when it belongs to. So we assume that maybe because of that mutation, the red banana, the anthocyanin pigment pathway is activated in red banana. So that we published that paper in Plant Cell Mutation Reports. And now the another aspect of the study conducted by the in our lab, that is the drought resistant genes occurring in banana. We have actually selected 2 cultivars. One is Amritasagar and another is Monthan. Because Monthan has, it is known that it has got more, resistance towards, I cannot say resistance, tolerance towards drought, whereas Amritasagar is susceptible towards drought. From this picture, you see, the Amritasagar is the 3 cultivars, where on the 10th day, if we don't give water, on the 10th day itself, the plant started the leaf started yellow in color and the leaf started drying out. In the 20th day, you can see all the leaves in the bottom is dried out. Whereas Monthan, in the 20th day, actually the yellowing starts. That means that in Monthan, more time is taken for the drought stress response. So it has got more tolerance towards drought. So we actually selected these 2 things, and we selected about 18 genes which is known for drought resistance, and then did real-time PCR analysis, see how these genes are being regulated in these 2 cultivars. You see this is the summary of the work done in that time. We can see that in Monthan, upregulated one gene, specifically one gene, that is MA means MUSA, SPTK PBS gene, that is upregulated, whereas in Amritasagar, which is susceptible to drought, that specific gene is downregulated. And whereas in MA, MUSA acuminata, SPS gene, which is downregulated in Monthan, is upregulated in Amritasagar. So there is variation in gene expression, in the class of genes which is controlling the drought resistance, there is variation. So now we are doing a little bit of proteomics, so we can actually isolate and classify more genes which is related to the drought resistance. So these are the people who actually worked along with me in this area. I have been working with Professor Pat Harrison and Dr. Trude Shawarkar, University of Leicester, UK. Well, a lot of molecular work has been done in the university there, along with in their own laboratory. And in university department, 2 of my PhD students, Dr. Smita and Dr. Reshmi, worked in banana for their PhD program as well as their PDF. So, most of the work done their PhD program in the department. So these, I'm really thankful for my student and as well as my collaborator, Professor Harrison and Dr. Trude. for helping me to realize some of the questions, hypotheses we actually identified in the beginning, and we came to some conclusions. All the work which I have presented here has been published in journals, and I have given all the publication detail also along with the presentation itself. And and now, for all the young students who is sitting there, don't you think that this is a challenging area where you can actually contribute a lot if you actually take up this time? There are a lot of things you have to work in banana again. For example, banana, the quality of the fruit, it is it beats fatigue, it fights constipation, it kills acidity, it maintains BP, it gives instant energy, and it gives you a glowing skin. So all these parameters belong to banana is make it as an ideal crop for more studies. And moreover, I would like to say a few things for example, just before me, we were talking about a very specific banana which belongs to only in Thrissur. But if you really want to improve the quality, we really want to know how these kind of things are working. Epigenetics is an area where a lot of work has to be done. Why the banana has got this nutritional quality? This specific character belongs to one particular cultivar, especially vitamin C content. If we can enhance the vitamin C content in the Cavendish variety, that will be a challenging question for the research community, because we have a lot of vitamin C in all this plantain variety. But the Cavendish, all this cooking desert banana, the vitamin C content is minimum. But we have got varieties in Papua New Guinea where we have got deep yellow colored banana which is rich in vitamin C. How can we actually upregulate the gene expression in Cavendish variety where the vitamin C content can be enhanced? That will be a challenging question that will be helping a lot of children because of the deficiency of vitamin C. So these kind of challenging questions are there in this crop, which the coming generation, the young faculty members sitting here, they have to take up and work on it. So I think this is why I took up this assignment of giving a lecture here, because a lot of students who is working in agriculture will be there, and I really want people to work on this crop, because this is really a good crop, which is the research community is well connected. You can actually have many more discussion with anywhere in the world, wherever you sit. People will communicate with you and share the data, so the research will be much easier way to do. So that is enough. Thank you for this opportunity. Thank you, the chairman. Thank you.